Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and we are into the revision mode. We are revising chapter number 8, human health and diseases, and this is meant for 20th October 2020. And in this session we are going to study very important, a 5-mark question repeated last one, life cycle of Plasmodium. This is a major question. And in this, the all important thing that one must note is the flow chart. You must practice this flow chart. So Plasmodium enters into human body as small sporozoites through the bite of infected female Anopheles mosquito. Here you must remember two things. Female Anopheles mosquito is a carrier or vector, one who carries pathogen. And who is the pathogen? It is the Plasmodium, which is a protozoan. Please remember this. Some people think that it's bacteria. No, it's not bacteria, it's protozoa. Plasmodium is a protozoan. Okay. So the female Anopheles mosquito, that too female Anopheles, female Anopheles mosquito carries this Plasmodium and multiplies within the liver cells. Now this is target number 1. Later attacks the RBC. So there are two areas in human being that are being attacked by the Plasmodium. One is the liver, second is the RBC, red blood cells. So this results in rupture of the RBC, breaking of RBC, and this rupture releases a toxic substance called hemozoin. Again, NEET exam, hemozoin, HEMOZYN, which is responsible for high fever, chills recurring every 3 to 4 days. These are the symptoms of malaria. So malarial parasite requires two hosts. One is human being, second is female Anopheles mosquito. So two hosts are required to complete their life cycle, and female Anopheles is the vector of this disease. And we must be thankful to Dr. Ronald Ross. So it is also known as Dr. Ross cycle, malarial cycle or Ross cycle. So thanks to him, he has saved the world. And this Ross cycle or the Plasmodium cycle or life cycle of Plasmodium or malarial flowchart, all these mean same. So how does it start? We take from the human beings. First, as you are seeing, female Anopheles mosquito bites a human being, and once it bites, what does it release from its saliva? From its saliva it releases hypnozoites, and these hypnozoites, after the bite, mixing with within the blood, enters into the liver. That's why we say hepatic dormancy. Hepatic cell means liver cells. So this is the first target, as I said. Once it attacks the liver cells, what happens there? The mature liver, the merozoites will be entering into each liver cell, and the liver cell will be blasted. So mature liver seizoant. And once these mesozoites break off, get released from the liver cell, now they travel in the blood and reach the erythrocyte. Erythrocyte is nothing but RBC. So it enters into the RBC now, and now it's called trypanozoites. And these trypanozoites keep on multiplying within the RBC, red blood cell. And once it is, this RBC cell is full, completed, now erythrocytic seizoant. So this seizoants break the RBC. As a result, rupture of the erythrocyte takes place. That is, RBC will be broken, will be blasted. And when RBCs are broken, now there are two cells that are getting broken. Look at this, liver cell is broken, RBC is broken. Now once this RBC gets broken, now the erythrocytes will be releasing a male gamete and a female gamete. So you get gametocytes. these gametocytes now will be entering into again female Anopheles mosquito because of the bite. So when it bites, the gametocytes enter into the mosquito and become microgametes, and sooner they become big enough, becomes macrogamete, and there is zygote formation. So zygote formation is taking place inside the female Anopheles mosquito, that too in the gut, that is intestine. So in the midgut of the mosquito, the zygote gets attached and becomes ookinete, and this becomes oocyst, and oocyst now bursts that itself. So the cyst gets bursted and a lot of sporozoites, these are the dangerous fellows, sporozoites are released, and these sporozoites enter into the salivary gland of the female Anopheles mosquito. So there they stay. So when the female Anopheles mosquito bites the next healthy person, these sporozoites are released into the healthy person, and the healthy person gets infected. So this is how the Plasmodium has got to have two hosts, that is, one is the human being, second is the mosquito. So two hosts are required to complete the life cycle. This is what Dr. Ronald Ross discovered, for which he was suitably awarded and recognized worldwide. So basically, what is required to overcome all the diseases, any disease, not only malaria? Immunity, the ability of the host cells to fight the disease causing microbes, and disease causing microbes are pathogens, and due to immune system is called immunity. There are two types of immunity. Innate immunity. Innate means within. Non-specific types of defense presents at the time of birth and provide different kinds of barriers to the entry of foreign agents into the body. That is, by birth we get this innate immunity, and it has got four types of barriers, means obstructions. So these four types of barriers will try their best to prevent the entry of a pathogen or any foreign substance. What are those four barriers that innate immunity possesses? One, physical barrier, first one, and skin is our BSF, border security force. It will not allow, the skin acts very tight and it does not allow any foreign substance. So that is our BSF. And then mucus, second one, coating of epithelium lining in the respiratory system, as well as gastrointestinal system and urogenital tract. So there is a liquid part, mucus part, which will also try to prevent any pathogen entering inside. And second one, physiological barrier. Acid, HCl, acid, it is produced in the stomach, okay, and that acid will also try to prevent the entry. And of course our own saliva in the mouth is good. And then third one is cellular barrier. So we have got leukocytes, neutrophils, monocytes. These are types of WBC. These WBC, who are the soldiers and scavengers of our body, will try and fight with the incoming enemy. And then cytokine barriers. Virus infected cells secrete a protein called interferons, and these interferons also will give its best fight to the pathogens. So basically innate immunity, and another one we said is acquired, one that we develop during our lifespan. So basically, what is the difference between the two, innate immunity and acquired immunity? It is present by birth, innate as the word refers to, acquired as it says develops during lifetime. Under innate, the immunity remains throughout life. This acquired one can be short-lived or maybe lifelong. Contact or exposure with pathogen or its antigen is not essential, there is no need. Whereas contact with the pathogen or its antigen is essential. Innate immunity is inheritable, generation to generation in the family. Acquired immunity cannot be passed on to the next generation except for a brief period to neonates, only brief period. And it protects the individuals from contraction of diseases of other organisms. It protects the individuals from pathogens present on other members of the same species. That means it is specific and for the time being. So acquired immunity, it is pathogen specific, as I said just now. Pathogen specific defense characterized by memory. This is important. So everything is recorded in our brain, and whenever we are attacked frequently by the same pathogen, our memory helps us to fight easily. When our body encounters a pathogen first time, produces a response called primary response of low intensity. Subsequent encounters by the same pathogen produce highly intensified response. This is called secondary response or anastic response due to memory of the first encounter. This is based on memory. So primary and secondary responses are carried out with the help of B lymphocytes, bone marrow, and T lymphocytes, thymus lymphocytes. B lymphocytes produce army of proteins called antibodies, anti against body. Of course, you know, each having 2 light and 2 heavy chains that we'll see in the structure of the antibody. It involves 2 types of lymphocytes. B lymphocytes show humoral immune response, so humoral immune HI, and T lymphocytes show cell mediated immunity, CMI. And in general, what is the structure of the antibody? This is how the antibody will be there. One long arm right from here to here and one short arm. Okay, so the antibodies are protein molecules. Of course, we know they are protein bodies, and they are called IgEs, immunoglobulins, and are of various types like IgA, immunoglobulin A, immunoglobulin M, IgE, IgG. Each antibody molecule consists of 4 polypeptide chains. So you can see 1, 2, 3, 4 polypeptide chains. 2 are long, called heavy chains, and you can see 2 and 3, they are long, so they are heavy chains, and other 2 like 1 and 4 are short, they are called light chains. So you got 2 heavy chains and 2 light chains. Both are arranged in the shape of Y, alphabet Y. Hence, an antibody is represented as H2L2. H stands for heavy chains 2, L stands for light chains 2. So you can see here there is a 2, the light chain. You can see another light chain this side, heavy chain here, variable region of heavy chain here, and this one more heavy chain. And what is important is antigen binding sites are there. Look at this, antigen binding sites, and there is variable region of light chain, constant region on light chain. So there is variable region and constant region on both the chains. And then it is basically these immunoglobulins are having disulfide bridges, sulfur bridges will be there. So basically an antibody, remember, should have H2L2. On the basis of production of antibodies, immunity can be further categorized as active immunity. Body produces its own antibodies against antigens. Second, passive immunity. Ready-made antibody is transferred from one individual to another. Okay. And then colostrum, third one, contains antibodies IgA. Again, NEET exam. Specific antibody is IgA in the colostrum. It's an example of passive immunity. Mother is giving to the baby through milk. That's why it is called colostrum. Passive immunity provided by the mother to a child. So different types of antibodies produced in blood include IgA, IgM, IgE. They are called humoral immune response due to presence in the blood. So human immune system can distinguish between self and foreign molecules or foreign bodies. Sometimes due to genetic or unknown reasons, the body attacks the self cells. This is unfortunate. This results in damage to the body and called autoimmune diseases. So one of the worst things that human beings experience is rheumatoid arthritis is due to this failing of knowing oneself. And then of course allergies, the exaggerated response of immune system to certain antigens present in the environment is called allergy. Like all of us must have experienced one time or the other in our lifetime that we are allergic not only to some people, to certain substances also. Like somebody is allergic to brinjal, somebody is allergic to the seeds, for example parthidium seeds. Somebody is allergic even to face powders, somebody is allergic to lipsticks, somebody is even allergic to gold. So what is exactly happening is the person who is allergic to particular substance, and when that particular substance is either taken in or applied outside, our body shows immediate response in the form of red pimples coming out throughout the body and lot of irritation in the body. So that is exaggerated response of immune system. The substance to which such immune response is produced is called allergen. The substance which is causing this allergy is called allergen. So allergen can be a face powder, allergen can be a cream, allergen can be a vegetable, allergen can be a dress even like silk shirts, allergen can be gold, and most of the time sometimes the allergy can be from environmental sources like as I said, Congress grass, the seed of the Congress grass, Parthenium. So the antibodies produced due to these are called IgE. Again, NEET exam, remember, for allergies, which is the antibody. So antibodies produced due to these are IgE. Allergy is due to secretion of chemicals like histamine and serotonin from the mast cells. So we'll continue to talk about various parts involved in the immune system in our next session. Until then, keep on practicing.